Assalamualaikum and hi everyone. Today we will be presenting our research article titled Metabolomic Changes in Renal Cell in Response to High Glucose Exposure based on liquid or gas chromatography coupled with mass spectrometry. Before we go into the detail of the paper, allow me to briefly explain what is this research is all about and how it relates to our study metabolomics. This article focuses on how kidney cells respond metabolically to high glucose exposure, which are a condition that stimulate diabetes. Instead of using animal or human subject, which can be influenced by many factors like diet, genetic, and environment, the researcher used in vitro cell model. This approach provides more control and reproducible conditions, making it easier to identify the disease-specific biomarker. What makes this study interesting is the use of dynamic metabolomics, which tracks how metabolites levels change over time during the disease progression. The researcher combined this approach with advanced statistical tools and two powerful analytical techniques, which are the liquid chromatography mass spectrometry and gas chromatography mass spectrometry. These methods are well known for their sensitivity and ability to detect a wide range of metabolites with high accuracy. Through this tool, the study was able to identify the potential early biomarker for dynamic neuropathy, which is a serious kidney complication caused by diabetes. This highlights how metabolomics can contribute significantly not only to early disease detection but also to monitoring disease progression and supporting personalized treatment strategy in the future. This article is closely related to our own studies in metabolomics, especially in understanding how small molecule or metabolites changes in cell, tissue, or fluid in response to external stimuli or internal genetic alteration. Overall, this paper shows the real-world application and importance of metabolomics in modern biomedical research. Objective of the research. First is to establish a dynamic non-targeted cell metabolomics platform using the LC-MC and GC-MC for analyzing metabolomic changes in renal cells exposed to high glucose. Second is to investigate metabolomic alteration in two types of red kidney cells under high glucose condition at different time points. Next is to identify the potential metabolomic biomarker related to the development and the progression of the diabetic neuropathy. Lastly, to validate the selected biomarker using the clinical plasma sample from healthy individuals, diabetic mellitus patients, and diabetic neuropathy patients. For the methodology, in this study, two types of red kidney cells were used, which are the renal tubular epithelial cell and glomerular mesenchymal cell. These cells were cultured and then exposed to two different glucose concentrations, which are a normal, low glucose condition and a high glucose condition to stimulate the diabetic stress. The exposure were conducted at 4 different time points, which are 12, 24, 36, and 48 hours, to observe the dynamic changes over time. After the treatment, the cells were harvested and rapidly quenched using the liquid nitrogen. This step stopped all the metabolic activity to preserve the metabolites at the moment. The cell then was subjected to solvent-based extraction to isolate the small molecule. Next, the extracted metabolites were analyzed using two powerful analytical techniques, which are the liquid chromatography mass spectrometry or LCMS and gas chromatography mass spectrometry or GCMS. These instruments help to separate, detect, and measure a wide range of metabolites in the sample. After data collection, statistical tools like principal component analysis (PCA) and partial least square discriminant analysis (PLSDA) were used to identify the pattern and distinguish between the low and high glucose treated group. Finally, to validate their findings, clinical plasma samples were analyzed. These samples come from 55 healthy individuals, 103 patients with diabetes, and 57 patients with diabetic neuropathy. This step helped to confirm whether the metabolic changes seen in the cell model were also present in actual human cases. For the result demonstrate that dynamic metabolomic profiling of renal cell under high glucose stress can reflect disease-related metabolic shift. The researcher found that the palmitic acid and linoleic acid can be identified as potential biomarker for the progression of the diabetes neuropathy. Valine, leucine, and isoleucine found to be elevated in diabetes mellitus patient but decreased in diabetes neuropathy, suggesting that they can serve as early warning biomarker for kidney damage in diabetes patients. Lastly, clinical validation of metabolites biomarker for diabetes neuropathy. In this study, a total of 215 adults were recruited from the second affiliated hospital of Zhushu Medical University. They were divided into three groups. The first group was the healthy group, which consisting of 55 individuals. The second group was the diabetes mellitus group, were consisting of 103 subjects. And lastly, the third group was the diabetes neuropathy group with 57 participants. According to the research, the investigator found several key changes in metabolites level. First, palmitic acid will increase in individuals with diabetes and even higher in those with diabetic neuropathy. This suggests that palmitic acid could serve as a potential biomarker for early detection of kidney damage in diabetic patients. In contrast, linoleic acid and stearic acid were found to be lower in patients with diabetic neuropathy. This indicates that fat metabolism is disrupted as kidney complications develop. The researcher also examined three amino acids, which are the valine, leucine, and isoleucine. They were elevated in diabetes but decreased again in diabetic neuropathy, showing a distinct patient that may help us to understand the disease progression over the time. In summary, the study highlights palmitic acid and linoleic acid as promising biomarker for diabetic kidney disease, while the amino acid could help in tracking how diabetes progress to kidney complications. Importantly, the pattern observed in both cell metabolomics and clinical plasma sample were consistent, supporting the biological relevance and reliability of these findings.
Hi everyone, today I will be presenting the principles and application of two important analytical techniques in metabolomics, LCMS and GCMS, based on the study published in Frontiers in Pharmacology 2019. The study focuses on identifying potential biomarkers in diabetic nephropathy using these techniques. This research applied non-targeted dynamic metabolomics to investigate how renal cells respond to high glucose conditions. Two cell lines were used: renal tubular epithelial cells and mesangial cells. The study employed LCMS and GCMS to analyze the metabolic change and identify biomarkers related to diabetic nephropathy. LCMS stands for liquid chromatic coupled with tandem mass spectrometry, while LCMS stands for liquid chromatograph coupled with tandem mass spectrometry. In this method, compounds are separated in liquid phase using chromatography. They are then ionized using ionization. A quadruple time of flight mass spectrometer detects and fragments ion based on their mass to charge ratio. This allows high resolution detection of complex and polar metabolites. GCMS stands for gas chromatography mass spectrometry. This method first derivatives and vaporized samples for separation in a gas phase. It uses electron ionization to ionize volatile compounds. A single quadruple mass analyzer then detects on ions based on mass to charge ratio. GCMS is ideal for analyzing volatile and also thermally stable compounds like fatty acids and sugar. In this study, both LCMS and GCMS were used complementarily. LCMS helped identify changes in amino acids like leucine, isoleucine, and valine, purine metabolites like hypoxanthine, and lastly, glycerophospholipids and sphingolipids. GCMS focused on fatty acids such as palmitic acid, stearic acid, and also enolic acid, carbohydrate like glucose and galactose. Through dynamic metabolite profiling, the study identified palmitic acid and linoleic acid as potential biomarker for diabetic nephropathy, valine, leucine, and isoleucine as potential indicator of diabetes mellitus. The fluctuation in these amino acids also suggests they may predict the progression from diabetes to nephropathy. In conclusion, LCMS and GCMS offer complementary strength in metabolomics. LCMS is suitable for polar complex metabolites, while GCMS is ideal for volatile low molecular weight compounds. Their combined use in this study enabled the discovery of novel metabolic biomarkers with potential clinical application in the early detection and monitoring of diabetic nephropathy.
Next, for this part, I'm going to explain the advantages of method used in the study we reviewed. First, it's very sensitive and can detect many types of metabolites. The tools like LCMS and GCMS are used and they can detect small molecules, for examples like amino acid, sugar, and fats. This is really useful because the small molecules can tell us about what is happening inside the bodies, especially if there's a disease. So the data collected is clear and really helpful for finding biomarkers. Second, the method is easy to control and repeat. The researchers use specific kidney cell lines and RK52E and HBZY1. These cell lines make it easier to control the conditions in the lab. So outside factors like age or diet do not affect the result. This means the experiments are more consistent and the results are more reliable. Lastly, it allows us to study changes over time. In the article, the authors observed the metabolites at 12, 24, 36, and 48 hours. This step-by-step observation helps to see how the cell respond to high glucose at different times. So we can really understand the process, not just one moment. So overall, this method is very helpful because it gives clear results, is easier to repeat, and show how cells changes over time. Now, let's talk about some of the disadvantages of the method based on the article we reviewed. The first one is the cell models do not fully show how the whole human body works. In the research, the authors use kidney cells in the lab setting, but these cells are not as complex as real human organs. So even though we get some good results, it might not fully reflect how the entire body would react in real life. This one limitation we found. Next, even though the method is easier to control and repeat, there's still a risk of losing some metabolites. This can happen during quenching steps. That's the part where the chemical reaction are stopped quickly. If this step is not done properly, some important metabolites might be lost. This can affect how accurate the results are, so it's something researchers need to be careful with. Lastly, the biomarkers found may not be kidney specific. In the study, biomarkers were found in plasma, but plasma contained molecules from the whole body. So we can't be sure if the changes seen are really from the kidney only. This makes it harder to tell if the kidney is the main source of those biomarkers. In our review, we found that the method used by the authors had a limitation. They only used kidney cells in vitro, specifically NRK52E and HBZY1 cell lines, rather than using whole animals or human models. According to the authors, in vitro studies are cheaper and easier to control. However, they do not reflect how the whole body would respond, since organs and systems in real life are connected and interact with each other. This means the metabolic changes observed in isolated kidneys cells might be different from those in a living human. So the finding may not fully represent what would happen in real patients. That's why we think this is an important limitation to consider when interpreting the results.